and we talked about how you're going to isolate the genomic DNA, the solutions that you're going to use in pursuit of isolation of the genomic DNA. So in your intro, you want to make, and this is going to be a long intro because you have 5 different topic areas to talk about. You want to make sure you talk about the isolation of the genomic DNA, why the genomic DNA is going to bind to those magnetic beads, how those magnetic beads are separated over the side to make removal of the supernatant much easier, why the DNA comes off of the magnetic beads, everything that I talked about in lecture. So please make sure you're looking at the protocol when doing that. You need to discuss oligonucleotide synthesis. And so remember, you have that page showing, which I think is actually page 99, which is why there's like a little arrow, because that's just the first page. Remember that you have 5 steps, and you have a chemical that's used in each step, and you have a purpose for a chemical for each step. So you want to talk about that in your intro. You want to make sure you talk about agarose and acrylamide gels. Are we using native? Are we using denaturing? How do they differ in terms of what they can separate? Things like that. Make sure you're talking about PCR. There's actually 2 different PCR experiments in the book. The first one is better because it talks, which I think is on page 119 or something. The first one talks about the actual ingredients that are put into it, and that you could use to refer back to my lecture. But the second one is we're using like a master mix with a buffer, with the nucleotides and everything all in there. And then so we're just using this master mix, and we're sticking our DNA in there. So the second protocol is not as illustrative in terms of all the solutions that are used in a PCR reaction. So you might want to refer back to the earlier experiment for how things used to be put together, all the things that go into a master mix type thing in a PCR reaction. And then we ended up talking about short tandem repeats, and we left off with talking about our short tandem repeats. And I wanted to remind you here that we had talked about when you have genomic DNA, you can do PCR reactions on and this is the same thing that I showed you in class a week ago because it's spring break. You can have 7 and 8 repeats. Why is this not working? Let's try a different one. There we are, it's a little bit better. You can have 7 and 8 repeats on one chromosome, and you can have 7 and 8 repeats on a different chromosome, but you can still separate them on a gel because the PCR primers can be placed farther away from the area of repeats, or they can be placed closer in to the areas of repeats. So the fact that you have the same number of repeats at these genomic locations does not mean that you have to have completely different numbers of repeats to be able to separate them. By placing the primers at different locations, you can have them all on different places in a gel. So just as a reminder, this is what we had talked about in class. And so remember that these under the L right here, these are the markers that are telling you how many repeats you have. So if person number 6 right here, so there's 6 people, 1, 2, 3, 4, 5, 6. So if person number 6 right here has a specific banding pattern at CSF1PO, and has a specific banding pattern at TPOX, and a banding pattern at THO1, and a banding pattern at VWA, the VWA being the smallest in size because it runs the furthest on the gel. The primers just happen to be closer to the repeats here, and the primers are much farther away from the repeats here. But you'll notice both THO1 and TPOX and CSF1PO, they have almost overlapping numbers of repeats for their different size ranges. but you can still separate them all on a gel. So if you had VWA, for example, this would be 11 repeats. There's a 12 right there. 13, 14, 15, 16, 17, 18, 19, 20, 21. We're going to practice this again. So person number 6 at VWA, what would their number of repeats be in their DNA at the location VWA?
15 and 17.
Okay, not 16, 17, but 11, 12, 13, 14, 15, 16, 17, 18, because this band lines up with the 16, this band lines up with the 18, 19, 20, 21. So the person number 6 would be VWA 16, 18. And they're put in the computer like that. And kind of like an Excel spreadsheet is searchable when you have them in a large database that's called CODIS, that's run by the police department. CODIS stands for Combined DNA Index System. And you can have local CODIS, or you can have national CODIS database entries. It's searchable, and you can search to see if this person with VWA 1618 and TH01, this band, and TPOX, this band, CSF1PO, those bands. So when you have their complete genetic profile, they're put into the database, and then you can query the database and say, does anybody else have this exact same pattern? You can also query the database and say, does anybody have a close pattern? So, maybe if there's 13 different locations, so just again as a reminder, there's original 13 different locations, and the locations that they were on the chromosome. We talked about this a week ago. And if you have a close family member, sibling or parent, something like that, they're going to be genetically related to you. So one thing that the police department does is called familial DNA searching. And this was how they got the Grim Sleeper killer in Los Angeles. They had a DNA sample from the murder, and they put it into the database, and nobody matched. And then they did familial DNA searching at like 9 out of 13 locations, and they said, okay, someone is fairly closely genetically related to this person. This is before everyone did genetic genealogy. So they then started looking at, okay, so there was a brother who'd been in prison, which is why they had a partial match. And so they looked and they said, okay, someone is related, not this guy who's been in prison, but someone closely related to this person is the one who carried out the crime. And they looked at the son of this person. He was far too young to have carried out the crime at the time. I think he was like 3 when the crimes happened, so they said, okay, not him. And they looked at the brothers of this person that they had identified and said, well, we can't, sometimes they surreptitiously collect DNA. If they need to, they will follow someone. Police have, if they wanted to collect a sample, they wait until they see someone spit on the sidewalk to throw a piece of gum away. They've followed them in fast food restaurants to collect a straw that they have been sipping at from their drink. They will do lots of different things. They don't have the ability to get a warrant because they don't have probable cause, but anything which you discard in the trash can be legally collected, and then they can say, okay, we've collected this genetic material from this person, and now we can test this in our lab. And finally, they found not the brothers, but it was the father of this one guy that was in the system who turned out to be the one that had murdered a bunch of people in Los Angeles. So that's how the police department will use short tandem repeats to either be able to say, you are already in our system, maybe you committed a burglary or something like that, and now you're up to homicide and we find you. Everyone currently, the law is that everyone who is arrested for a felony has to submit a DNA sample into the system. And if you are found not guilty, or if the charges are not pressed for any reason, you're supposed to be expunged from the system. But California says, sure, we'll take you out of the system as soon as you request it. People don't know their rights under the system, and they fail to request it, so their DNA will remain in the system that will be searchable for any potential future crimes they or even family members might commit. So if any of you ever get arrested, make sure you request that they take your DNA out of the system. My DNA is in the system because I did an internship in the forensic biology at the San Diego Police Department. So as part of that, you have to submit a sample of your DNA in case you contaminate something in the crime lab. And I'll talk about contamination in just a minute. So here's the original 13. Now they're doing 24 different locations on the DNA with the accompanying statistics that go along with those. So let's start talking about, so this is just...
Wait, Kathy, 1 second.
Yeah.
Going back to this page,
yeah.
Each panel is one single gel. So I can orientate myself. B is a single gel.
Yeah. Oh, no. So B and C are actually the same gel, but they have different color markers, which is where I was going with this that I was just putting up right here. So just as a reminder, this says 16, but it's actually 24. Markers can be PCR'd in the exact same tube at the exact same time. So you've seen how 4 can be done and separated on a gel, but what you can also do is, in addition to having those 4 that are separated by size, you can have different fluorescent tags on your PCR primers. So you can have red or yellow or blue or green tags, and when you have those different colored tags, now you can tell the computer, show me only the red or show me only the green, etc. So that's how we're able to get all these different that's how we're able to have 24 now PCR'd all at the same time. And trust me, a lot of research has gone into this. You guys are thinking, oh, this should be pretty simple. You just mix all the primers together, but you don't want the primers to bind to each other. They have to have very specific concentrations. And if you don't get the concentrations of the primer just right, you're not going to get good PCR levels on all of the ones you're doing at the same time. So a lot of research has gone into exactly what sequences of the primers and exactly how much of each of these primers, because some sites are naturally going to PCR better than others. And I know this firsthand because I said, why should I be buying a $1,200 kit for my 567 class when I can just order the PCR primers myself and mix up all these and mix up 3 sets of PCR primers myself and do, with a enzyme and stuff like that, and do this for my class? Why spend all that money? Well, I tried it, and so with one concentration you'd get the TPOX to be really dark, and then you'd go, okay, well, I need to decrease TPOX and increase THO1 and CSF1PO concentration of primers. And then TPOX wouldn't work at all, and CSF1PO would work really, really well, and THO1 wouldn't work at all. And I tried. Finally, I said, fuck it, I'm buying the kit. So that's where all the money goes to when you buy a lot of these kits for this class, for the police department, is all the research that has gone into how can we make 24 different PCR reactions happen at the exact same time so that we get good amplification at all of these 24 different sites we're looking at on the DNA. So, as I said, this is called multiplex PCR, and so if you look at it, these would be, if you said to the computer, show me only the ones that have red tags on the primers, it's going to show you panel C. And panel B, for example, is the exact same gel as panel C, except all of these size ranges now, instead of having a red tag, have a green tag on them. And you have the ability to say to the computer, because it's all done and scanned on a computer that's going to read all of these fluorescent markers, so you can see what it looks like. In one lane you can have red and green. And so actually in one lane you can have red and green and yellow and blue and all the other different colors that they use that can be detected by specific wavelengths designed to pick up the fluorescent tags here. So if you are looking at VWA, we already said that was a 1618, and in class last week we said THO1 was a 6-8, and we said this is what's called homozygous, that you have two different sizes that can be resolved from one another because they are different sizes. There are 8 repeats here and 6 repeats here. So the one with 8 repeats is a larger sized fragment than the one with 6 repeats. And the primers are in the exact same location for both of those, but if someone at the position in THO1 in their genomic DNA has both a 6 and an 8 repeat at that location. And as a reminder, why possibly 2 different sizes? Because they have a mother and they have a father, and they've inherited the chromosome from their mother and their father. So they can have inherited a 6 from their mother and an 8 from their father, or an 8 from their mother and a 6 from their father. So there's 2 different ways that they could be a 6-8. And that's going to play into when we do statistics. So that makes a difference. On the other hand, our TPOX only has 1 band here. What did we call it when there's only 1 band? Homozygous versus heterozygous. Homozygous means there's only a single band. And so if you are an 8-8, what does that say about what you inherited from both parents? That you inherited the same 8 from both parents. And just as a reminder again of the lecture, that there are statistical probabilities of being a 6 or an 8 or an 8 here or a 16 here and an 18 here. And you can do those statistics to be able to say what is the possibility that anyone is going to match this pattern. So let's talk about this, the statistical probabilities. So there are probability tables, and in a minute I'll talk about the probability tables that are in your lab manual. But here is the probability table for VWA right here. So a company, and you don't have to worry about taking a picture of this necessarily, other than you want to know this right here. That's the important part right there. Companies, Promega and Agilent and things like that, have done lots of testing. They've gotten blood samples from a number of people. They did statistics and said, okay, what is the probability at the location VWA of having 14 repeats, or 15, or 16, or 17, or 18, etc. So they said, what is the probability of having each of these possible numbers that we see in the gel? And so 13.1% of the people had 14 repeats. 8.2% of the people had 15 repeats. 21% of the people had at least one 16. Now they may have had a 16 and an 18, but they had at least a 16 somewhere. So that they're not all going to add up to 100, because you have 2 different possibilities. Okay, so we said that our VWA person number 6 was 16 and an 18. So the probability of being a 16 is 21.1%, or 0.211. The probability of an 18 is 20.2, or 0.202. So the probability of being both a 16 and an 18 is 2 times the probability of the first times the probability of the second. This is just basic statistics. So 2, because there's 2 different ways you can be a 16 and an 18. 2 times the probability of the first times the probability of the second is 0.085, or 8.5% of the people have both a 16 and an 18 at VWA. So already we are, we are less than 10% of the people, and we've only checked one location on the DNA, and already less than 10% of the people are going to potentially have that pattern. And remember, we're checking 24 different locations on the DNA. So let's talk about the next one we're going to do. So THO168, we said that there's a 6-8 THO1. So this is the probability table for THO1. So the probability of being a 6 is 0.237. The probability of being an 8 is 0.117. And the probability of being both a 6 and an 8 is 2 times the probability of the first times the probability of the second. Doesn't matter which one you put first, you're multiplying, so you're going to come out with the same number, or 0.055. So 5.5% of the people are going to be both a 6 and an 8. What if we'd had only 8? How would you do the math there? What if we had only 8? Yeah, you'd square it. So 0.117 times 0.117. Why do you not need a 2 in front of that one? Because there's only one way you can be an 8-8. You had to get an 8 from your mother and an 8 from your father. So there's not 2 ways that you could be an 8-8. There's only one way that you could be an 8-8. So we've said now we have those probabilities for both. And to say, so what is the probability that a person is both a 16-18 at VWA and a 6-8 at THO1? So you take your probability of the first times the probability of the second, and now we've only checked 2 locations on the DNA, and already we are at less than 0.5% of all the people in the world are going to have that particular pattern, and we've only checked 2 locations. So when they start checking the minimum of 13, they used to only do 13, but if you take the most common numbers that are the potentially the highest probability for each one, 20%, 22%, etc., you get a number like 1 in 10 billion. Well, if you have a number like 1 in 10 billion, the population of the planet is approaching 1 in 10 billion, and you're going to have some smart defense lawyer, and you're going to have some jury who knows nothing about math and probabilities, and that lawyer is going to say, the probability is 1 in 10 billion, and the population of the planet is almost 1 in 10 billion. Therefore, there is another person that has this exact same pattern as my client, and that, ladies and gentlemen of the jury, is the person that committed this crime. So not that they're in San Diego, but they're somewhere in the world that they could potentially have the same pattern, and they would try to convince the jury that that person must be the one who magically came to San Diego and committed the crime. And if you have a jury who does not know anything about math, probabilities, science, they're going to believe it. Because lawyers will very often exclude anyone with a scientific background from the juries. They don't want anyone who knows anything about science that could potentially, in the jury room, explain to the other jurors how it really happens, etc. Or put their own spin on things. They don't want anybody. So when my husband was working at UCSD, he'd get called to a jury. Boom, he's the first one off because he has a master's degree in chemistry. They don't want anybody, who knows anything about science on a jury. So because of that potential problem of 1 in 10 billion, they said we need to do more locations on the DNA, and so they started testing more locations on the DNA. Some of the locations, more of the locations actually overlap with what Europe is doing, because Europe selected completely different locations on the DNA to look at than the United States did. Well, not completely different, but some differences. And they wanted to be able to exchange DNA data with Interpol. And so when they expanded to 24, they chose some of the locations that Europe was doing as useful. Now, remember that you have potentially tens of thousands of these different locations on the DNA that they could have chosen, but not all of them have good locations where everyone has this exact same sequence outside of the repeats, because that's one of the things they had to have. They had to be able to use one set of primers for everybody that would amplify for everybody. Not everyone has a wide enough variability in statistics. If everybody had, say, either 6 or 7 repeats, it's not going to be very useful for getting those numbers lower and lower and lower. So that's not going to be as good of a location. So they chose the ones that were best scientifically for use. And so basically they have numbers like 1 in 500 trillion that anyone would have the exact same pattern that we find in the DNA here. Now, one of the things they have to worry about in the crime lab, as I said, is contamination. And they take great pains in forensic biology to avoid contaminating the DNA sample. So when there is a crime scene, in the days of O.J. Simpson, you'd have people just walking all over the crime scene. You'd have firefighters coming in to see if there was any life in the body, which you have to do. You have to protect the person before the crime scene. That's just basic humanity. But once it's determined that the victim is deceased, then they back off. But in the case of the O.J. Simpson, you had criminalists, you had reporters that were on the scene taking photos, you had far too many police officers walking around the scene, things like that, potentially contaminating it. Then, when you find a sample that you want to take back to the crime lab, they have to collect a sample. They're taking photos of it to make sure that they're documenting where they found the sample and how much of the sample they found. Was it a small blood stain on a fence post, as was the case with one of the samples? Was it a blood sample that was taken from a suspect? Was it a sock that had blood on it? Things like that. So they're saying, here is a photo of what we've found, and then they're saying, okay, we need to collect it properly. So when you are collecting it properly, you're not just going to throw it in a paper bag with other items of evidence. You need to avoid any cross-contamination. So everything gets packaged separately with really good labeling saying who has had access to this package since it was collected at the crime scene. And so there's actually a label on the package that will say, okay, so here's the criminalist that collected it, and then they turned it into the evidence room, and then the evidence person turned it over to this criminalist in the lab, because despite what it shows you on television, the person that collects it at the crime scene is generally not the person that's going to be analyzing it in the lab, despite what CSI and stuff like that show you. And it also takes a lot longer than they show you on CSI. It's not a 1-hour process. So there has to be adequate documentation. And when you are a criminalist who's checking it out from the evidence room, for example, you are never breaking any of the original seals. You are doing extensive, because I know there's some people here who are interested in forensic science. You're doing extensive documentation on exactly what does the package look like, who's had it before you, where are the seals, you're taking pictures of it. All this is going in a notebook which can be subpoenaed for use in court. And so you better have your facts correct, and your notebook better be accurate, because when you're testifying in court, it might be 20 years later that you're testifying in court, and you're not going to remember what you did 20 years ago on any particular case. So all you're going to have to go by is your lab notebook and all the notes you took at the same time of your techniques and things like that. There was recently a case a couple of years ago, a case in San Diego where back in the day, back in the 1980s and stuff, when people were trying to say, was there sperm found in this situation. There are chemicals that you can use. It's called Christmas tree stain because it'll stain the different parts, red and green. But they needed a positive control. And it was very common for criminalists to use, male criminalists, to use their own semen. So they would ejaculate and they would take a sample of their semen and they would put it on a whole bunch of different little just pieces of paper, and then when a criminalist was working a case, if they needed to have a positive control, they would just take out one of these samples. So it happened that there was a cold case, and so there's a cold case squad in San Diego, and they were going back and they were looking at older cases that happened in San Diego, and they said, hey, we've gone back and we've done DNA sampling on this case, and we found a few sperm cells in this evidence. Now keep in mind, this was from the 1980s, well before DNA was regularly used as something that they would isolate, at a crime scene. They were just looking for, was there sperm in a rape case, was there sperm so that there's evidence of rape, kind of things like that. So they found sperm cells in this case, I think like 6 or 10 individual sperm cells on some of the items of evidence. This was also far before they worried about how much contamination are we going to have from one sample to the next. What they do now is, between samples, they're going to bleach the entire area. They're going to put down fresh paper and take it up at the end so that they're minimizing any potential contamination. They're wearing gloves. They're wearing a full face mask. They're taking great pains to avoid contamination in the crime lab. One thing I learned when I did my internship in forensic biology is about the one-way flow of information. So when they isolate DNA, they have a room for bringing the crime scene samples in, isolating any potential samples in that room, and it goes into an epitube, and the top of the epitube gets closed, and only that tube goes on to the next part of the lab. And the evidence then goes back to the evidence room. And from that tube, then they're isolating any potential DNA from that tiny little cutout piece of bed sheet if it's a rape case, or other thing that a swab if they had to swab, say, someone's breast in a rape case, if they think there was saliva or something like that. So whatever type of evidence they're trying to isolate DNA from. And when they're isolating DNA, they are very, very worried about contamination. They only have one tube open at a time. You never ever have more than one tube open at a time. And you're not doing samples from this crime scene as well as samples from a different crime scene. You're not doing samples from the suspect at the same time you're doing samples from the victim because of the potential for cross-contamination. If you have a bottle of solution that you need to add to your Eppendorf tube, you open the bottle, you get a brand new tip. We haven't been really, using brand new tips in this lab for everything, but they do in the crime lab. We're not going to worry about it in this class because we're not doing actual crime scene samples, but they're going to use a brand new tip that's been autoclaved and is completely sterile, and they're going to take up some solution, and then they're going to put the top back on that bottle of solution, because you can't have two things open at the same time. And only after the top is on that bottle of solution, then you can open your one sample and put the solution in. And then you close the top, and then you get rid of the tip and get a new tip and repeat the process. Yeah?
When you're putting in the tip, is there a sterile environment? Are there like blocking when you put in the tip?
You're not in a completely sterile environment because you're not worried about bacterial contamination, but you are worried about, you have a face mask and you have gloves and things like that. So, talking about the face mask, when I was beginning in the crime lab, and so they explained to me about the one-way flow of information and about how all the steps they took to avoid contamination, And so they were explaining the process to me, and after they explained the process to me, the guy that was showing me turned around and started doing it. And then I had a question, and he said, and he stopped what he was doing, and he turned around and he put up his face mask and he answered my question. And he said, okay. And I said, okay. You know, and then he put the face mask down, started doing, and then I had another question. He stops what he's doing and, you know, turns around, puts up, and I realized I'm pissing this guy off because every time he starts working, I come in with a question and he has to stop what he's doing. So they're not even talking while they are doing a sample because of the potential for contamination, because everybody spits a little bit when they talk. They're talking a lot, and particularly if you're trying to project your voice or something like that, and you might accidentally get some of your DNA sample into a crime scene sample. So back to the case that they found a couple of sperm cells in. So they isolated those few sperm, got a DNA profile off of them, and found that it belonged to a former criminalist in the laboratory. And so then they started questioning him as a suspect in the case. He tended to have a nervous disposition in the first place. So when they started questioning him, of course, he gets even more nervous and sweaty and stuff like that. So they're thinking, oh yeah, we've got our guy. So one thing that the police do is they tend to read body language and assume that if you have a particular type of body language, that you must be guilty. Police officers sometimes are not very good at looking at the evidence and saying, you know, what can the evidence show us? Where can the evidence lead us? They will just sometimes make assumptions as to who they think is guilty and try to go with that. So anyhow, the criminalist assumed that they would know that everyone used their own sperm for samples. The police department did not know that, never asked him about it. Is there any other way in which your sperm cells could have come into into this crime scene sample? And so basically they were prosecuting him. They were going to prosecute him for murder when he decided to hang himself over in Cuyamaca State Park. But his widow was pissed, and she found that, and this was in, if you want to look up the whole story, this was in the Union Tribune about I think a month ago. So she got a big settlement from the city of San Diego, basically for messing up and not doing the case properly and not finding out when the detective in charge of the case should have known that or you know, people in charge of the crime lab at that time should have mentioned that this was a possibility based on how standard procedure was done in the 70s and early 80s before DNA became so common. But once DNA became common, they had to really watch out for contamination that led to the one-way flow of DNA. So as I've said, after the DNA is isolated in the lab, it's taken to another part of the lab where the PCR is set up. Once it's set up for a PCR, it never goes back to the original lab. And then that PCR sample is taken to a instrument that it's not actually a gel, but it's a very thin type of sort of like almost agarose material in a very thin tube that is used to, that can separate, and that leads to what you saw there. It's just sort of shown as a gel format rather than what each individual tube has. And so once it's been through there, even the paper never goes back to the original crime lab. And this was, again, was brought home to me when I was walking down the hallway, and every door was locked, so you had to have a key to get in. So I'm unlocking the door and I see this big stack of paper outside the door, and I figure, oh, someone just had their hands full, they had to put the paper down. And so I picked up the paper and I was going to be helpful, and I said, oh, someone left a stack of paper, outside of the lab. And everyone looks at me and goes, no, one-way flow of information, it can't come in the lab. And it's like, oh yeah, even the paper, okay. So I learned a lot doing my internship about the potential for contamination and all the steps they take. So a one-way flow of information using mask and gloves, making sure that every tip has been autoclaved and is brand new, that they're bleaching every bench between samples, that they're using paper, that every sample they're using control, only one thing open at a time, and they have controls for every sample to measure the possibility of contamination. So when they have a batch of things to do a PCR on, they need to make sure that the PCR reaction works. So they want to have a positive control that can demonstrate that the reaction works, that the chemicals are all good. And they also need to have a negative control. And so in your experiment, when you're reading it, you will see that we're going to split it up. One group will do a positive control, one group will do a negative control. But a negative control is done to make sure that there's no DNA contamination in the solutions themselves. Because let's say a lab technician was making up a batch of solution and accidentally stirred it with their finger. Let's say they stirred it with their finger. Well, then they're going to have a bunch of skin cells in that solution, and any of those skin cells can then be taken up and will PCR just the same as a crime scene sample will PCR up. It does not take much DNA to get a genetic profile. It used to take, when they first started using DNA, they had to have at least a blood spot the size of a quarter. And then it became a blood spot, the size of the head of a pin. And then it became a blood spot the sharp end of a pin. They needed to have a very, very small blood spot. Now it's about 10 cells. You need to have 10 cells to get a genetic profile. And if you touch something, you're probably leaving 10 cells behind every time you leave a fingerprint somewhere. And they do what's called low copy number amplification. So there was a case in England where a child was kidnapped and later found dead. And as part of the crime scene, they took the little onesie that the kidnapped deceased child was wearing, and they decided they wanted to be able to isolate DNA from that. And so they did a DNA extraction protocol from the onesie, because not only would the mother, but potentially the kidnapper is going to have their genetic profile on that onesie if they picked up the kid. Well, they came up with a female DNA that did not belong to the mother. And so they found who it belonged to in England. I'm not going to go into the details, but they found who it belonged to in England. Well, and they arrested her in England. Well, it turns out she also had a small child, and she had been looking for onesies in the department store, and she'd been handling a bunch of the onesies, and just in handling the baby clothes, she was depositing enough of her DNA on those baby clothes that they were later able to, because the baby clothes were not washed after they were purchased. They just went straight on to being on the kid. So her DNA remained. So even though she had absolutely no connection to the kidnapping of this child, she became a suspect just because her DNA was found, and because they're doing this low copy number analysis of DNA. So contamination is an extremely important issue in crime labs. Okay, so we've talked about multiplying probabilities. Now, let's turn to page 139 in your lab manual. Page 139 in your lab manual. It's going to look kind of like this. So I realize you can't see the numbers unless you have your laptop open or something. Okay, so page 139 in your lab manual has the number of repeats right here. And so this is, on page 139, this is CSF1PO. It doesn't say CSF1PO, so just write page 139 CSF1PO. And it says, okay, so there's going to be 6, 7, 8, 9, 10, 11, etc. repeats. And there are different sets of numbers by that number of repeats. And they are Caucasian American, African American, Hispanic American, Asian American. because even though the probabilities are very, very close, they might potentially have slightly different numbers depending on a racial background, how somebody would identify as. So, for example, let me see if I can read the numbers here. At 12 repeats in CSF1PO, Caucasian American has a 30%, so 0.300 probability of having 12 repeats, but someone who's African American is 0.261, someone who's Hispanic American is 0.331, someone who's Asian American is 0.410. So slightly different numbers for these different numbers of repeats. And likewise, on page 140, and I also want to point out that CSF1PO, note that it goes from 6 to 15 repeats. So basically what you have here is 6 to 15 repeats. So this corresponds with this right here. That's on your page. So then we have on the next page of your lab manual, page 140, at the top is THO1. So the statistics for THO1, which go basically from 5 to 11 repeats. And if you look at your little green-white sample sheet I gave you, you'll see that the markers go basically from 5 to 11 repeats. Occasionally, for some racial groups, there might be a 4, but it's not at all common for the other groups because there is genetic drift going on all the time. And then lower are the numbers for TPOXX. So at the bottom of page 140 are your numbers for TPOXX that go from 6 to around, for the most part, 13, although a couple Asian Americans tend to potentially have a 14 and a 15. So these are where you're going to get your numbers that you're going to use. So what's going to happen in this experiment is you're going to, at different times for the 2 different times of lab, the 2 p.m. is doing it starting tomorrow, the 6 p.m. is doing it in a couple weeks, but you're going to do a buccal swab to isolate, to get your own DNA on a swab. You're going to go through the DNA isolation procedure that I wrote on the board over there, in the lab manual. And when you isolate your DNA, you're going to put it into a PCR reaction. Again, those page numbers that I put on the board, the PCR is going to be done with oligonucleotides, so that's why you're going to explain about oligonucleotides. At the end of the PCR reaction, you're going to be dividing your sample, and you're going to be running half on an agarose gel and half on an acrylamide gel, mostly so you can see how the different gels separate the different size ranges a little bit differently. All we really need to is, did you have some kind of a sample, and can we tell about what size it is? And then and what I said in lecture, we can't use your actual DNA to do statistics on, because that would require silver staining of a gel, which needs formaldehyde, and EH&S said we can no longer use formaldehyde with undergrads, which you are. So we have a pre-stained gel that has different people's samples on it, so you're just going to pick a person from that gel. So you're going to get the experience of PCR in your own DNA running in a gel, and then you're going to just look at a gel that we have pre-done with anonymous DNA samples on it that's already been silver stained, and then you're going to do the statistics on that. So you're going to basically say, okay, so let's say I pick person number 4, and person number 4 for CSF1PO, what is their pattern? For TPOXX, what's their pattern? For THO1, what's their pattern? Because those are the 3 locations we're amplifying the DNA at. So those 3 different locations we're amplifying. So you're going to then do statistics. You can do the statistics of, let's say I have, 16, well, that was VWA. Let's say I have 10 and 12 for CSF1PO. Okay, what's the probability of each? 2 times P times Q, then you're doing THO1 and TPOX, multiplying all 3 of those probabilities together to get a very, very low number. And at the end of your lab report, you're going to tell me what is the probability of anyone else matching your DNA. And so how you're going to do that is when you get that very, very low number, 1 divided by that very, very low number is going to give you a very, very high number. And that's going to be the probability that anyone else will match your DNA. Now we're only looking at 3 different locations on the DNA. So you're not going to get numbers like 1 in 10 billion. You're going to get numbers like 1 in 1,000 to 1 in 10,000. So that's the range you should expect. And so if you look at I've already told you what I expect for the intro. For the report, you're going to talk about contamination. List 5 things you could do to avoid contamination, which we did not do in lab just because of our lab constraints. You're going to analyze your PCR gel and talk about the banding pattern, the differences in agarose and acrylamide, and then you're going to do the statistics on it like we just talked about. Thank you. So a fairly simple, hopefully 1-page lab report. Like I said, the intros get harder and the reports get easier in this half of the semester. Any questions? Okay. That's all for me.
I have a question. So at the end, there's 4 different alleles in the thing, but we're only doing 3. What is the last one? Is that the VWA?
Just a different location on the DNA. It just happens that I picked a kit from Promega that amplifies 3 of those.